The overall goal of this experiment is to display our protocols for the culture and transfection of HEC293 and C2C12 cells with CatchER Plus and its application for monitoring ERSR receptor-mediated calcium transients and the measurement of the in situ KD.
This method can help answer key questions in calcium signaling and imaging, such as the basal calcium concentration and its rapid changes in the ERSR of different cells, and how to apply biosensor CatchER Plus for these measurements. The main advantage of this technique is that the fluorescence response of single cells can be recorded in real time with rapid kinetics.
The implications of this technique extend toward diagnosis of ERSR calcium-related diseases, because CatchER Plus can monitor abnormal calcium transients in these organelles that arise from dysfunctional calcium signaling elements. Visual demonstration for this method is critical as the cell transfection and fluorescence imaging steps are difficult to learn because cell culture and microscopy have multiple areas for error.
Demonstrating the procedure will be Florence Reddish, a postdoc from our laboratory.
To begin this experiment, seed HEC293 cells onto the sterilized glass microscope slides in the 6 cm dishes and culture for 1 to 2 days until they are about 70% confluent on the day of transfection. Next, empty DMEM from the dish and add 3 ml of the reduced serum medium. Then, add DNA transfection reagent.